Hi, my name is Grace, and today I'll be talking about my project. So, my project is about cell segmentation, and is specifically about a comparison between automated and manual cell segmentation. So, before I start talking about my project, it's probably important to give a little bit of background information. So, basically, what is cell segmentation and why is it important? Well, simply put, cell segmentation is the splitting of a microscopic image into many different segments. And in this case, each segment represents an individual cell. And cell segmentation is a fundamental step for performing any kind of quantitative single cell research. So this is because in cells, the location and the organization of organelles matters, and it will affect the potential function of the said organelle. And so we must segment the image into cell bodies to be able to accurately pinpoint the exact location of the organelles of whatever it is that we may be trying to analyze. And then once we have segmented these cells, we can now perform single cell analyses. So, for example, if we are trying to study, say, the gene abundance or expression levels of a gene in breast cancer, of a specific gene in breast cancer, using these cell images. So after imaging, we would have an image like this. And so to the computer, this is just, it hasn't been segmented. So it's just, it doesn't recognize that there are different cells. So even though maybe to us it looks like, for example, this could be a cell, this might be one, this might be one, the computer doesn't recognize it as cells. And so when we go to try and perform a different analysis, the computer can only give us data about the total transcript count of the gene, and we can't perform single cell analyses, which in this case is what we want. So then we have to go and segment the image, and we're left with something like this. And so here you can see that the original image is still on the bottom, except now we have these different masks over uh, each of the sort of little sections. And so now the computer and the algorithm can recognize that, for example, this is a cell, and this is another cell, and this is also another cell. And so now when we go to collect data, the computer will recognize that these are different cells, like I said, and then they can give us specific sort of single cell data, and then we can use that to perform whatever kind of analyses that we are trying to perform. However, up until recently, cell segmentation has mostly been done by hand, and this can be very costly in time because one has to be very careful, very precise when segmenting, so we don't accidentally leave something out or include something extra, because that will all affect your data. And so a recently developed program for automated cell segmentation is called CellPost. And like I said, CellPost is a fully automated algorithm that is capable of performing cellular segmentation. It does require a data set to train, but the program is already trained upon installation, so you don't need to worry about that. The program is based on deep learning neural networks and different vector gradients, and it can be easily retrained to segment a specific type of image. So for example, if you're working with tissue cells in particular, you can retrain it so that the program is better suited for segmenting that type of cell. And this will save time and also resources. So for my study, I wanted to look at how efficient and accurate automated cell post segmentation is in comparison with manual segmentation, because CellPost is such a new program. And also I wanted to look at the end of the day, is it really worth it to use automated cell post segmentation instead of just manually performing segmentation? So of course, with any scientific question, you have to perform experiments to collect results and perform analyses. So for my experiment, I used cellular images of RNA transcripts that were produced from 2 rounds of MERFISH imaging. With experiment R4, I used images generated from the first round of imaging. And for this experiment, the cells were non-gel embedded when we imaged. So that means that the cells are not as clear cut, and it can be a little bit harder to sort of distinguish where exactly one cell ends and another begins. And then with experiment R49, we use gel embedding. And so this makes it more clear and sort of more distinct where the cell boundaries are. So for my experiment, the control was the manually segmented images that I did for both experiments. And then the variable was just how well automated segmentation performed on fields of views, both with more distinct boundaries and with less distinct cell boundaries. So for reference, this picture on the left right here was an image of a field of view from experiment R49 that was performed with gel embedding. So you can see it's like much more clear where the cells are. So like these are pretty distinct cell boundaries. And then this image on the right was performed without gel embedding. And you can see it was just it's a little bit harder to tell. For example, you have spots like this or right here where where exactly does one cell end and the other begin. Okay, so for my results, so for experiment R49, this first table showcases the results of segmentation in terms of all the cells kind of grouped as a whole. And the second table down here kind of showcases the statistics for individual cells. So in the first table, we can see the total barcodes, which in this case essentially means the number of barcodes or transcripts that were assigned to a cell. So we can see that CellPost assigned many more barcodes to cells than manual segmentation did. And then we can see that that's also reflected in a lower percentage of barcodes assigned to cells for manual segmentation compared to automated segmentation. And then that's also kind of consistent throughout all of the data. We can see that CellPost kind of outperforms manual segmentation with both, like, for example, median cell size, genes per cell, cells per field of view, median barcodes per cell. It all kind of consistently performs better. And then also with experiment R4, we can see that it's kind of a similar story. It's very consistent. So once again, CellPost performs better in terms of including all of the barcodes, including more of the barcodes, not all of them. However, with experiment 4, there is a outlier with the median cell size. So in manual segmentation, actually, it produced a cell size that was greater than CellPost segmentation. And so this is probably because of a discrepancy between the way that a human eye and the way that the algorithm draws the cell boundaries. Because with CellPost, the algorithm basically computes the nucleus of the cell and then uses a series of mathematical calculations to determine the cell's boundary. But with manual segmentation, the human eye just looks for example, sections of the kind of lit up barcodes to group together. And so that is much less consistent than a series of mathematical calculations. And so as these results show, this method of sort of visually determining where one cell ends and another cell begins is pretty inconsistent from cell to cell. And it also causes some important barcodes to maybe be left out. And so that could also account for a larger median cell size because maybe we drew a larger cell boundary, but it didn't actually include more barcodes. So in conclusion, I found some pretty consistent trends in the results. So CellPost, first of all, consistently outperformed manual segmentation in both experiments in terms of accuracy. And it was also much more consistent than manual segmentation. And like I said earlier, this is probably because there's less cell-to-cell variation with automated CellPost segmentation because it is based on a mathematical algorithm. Also, I noticed that manual segmentation was more prone to outliers, and manual segmentation, of course, left out more barcodes than automated segmentation, and that led to a lower percentage of barcodes being assigned to a cell, which would then affect any data analysis that you wanted to perform on the data that you collected from your segmented images. So in conclusion, after my experiments, I deducted that cell post provides a more efficient, accurate, and consistent method of drawing cell boundaries than manual segmentation. It also saves time because multiple fields of views can be segmented at the same time. And cell post overall just produces results that are more beneficial and accurate than those produced by manual segmentation. And so cell post at the end of the day is worth it to use and is very beneficial. Thank you.